Assalamualaikum warahmatullahi wabarakatuh. Hello, everybody. I'm Gherda Febrianti from Biology of Rinci 2019, University of Malang. On this occasion, I will explain about cell divisions. Cell divisions, there are two, namely mitosis and meiosis. Before entering the process of mitosis preceded the process of interphase. In the phase, there are three stages. The first stage, G1, at this stage, cell is gathering energy. Second stage, S, occurs DNA duplication. Third stage, G2, ready to enter the mitosis process. Next, mitosis. The function is replace damaged cells, occurs in body cells or somatic cells, produce two sibling cells. Child chromosomes are the same as parent chromosome and consistent of four bases. The first phase of mitosis is prophase. The chromosomes appear condensed, and the nuclear envelope is not apparent. And next, metaphase. Thick, coiled chromosomes, each with two chromatids, are lined up on the metaphase plate. Next, anaphase. The chromatids of each chromosome have separated and are moving toward the poles. And then telophase. The chromosomes are at the poles and are becoming more diffuse. The nuclear envelope is reforming. The cytoplasm may be dividing. Next, meiosis. The function is reducing chromosomes, occurs in gamete cells, lasts twice, meiosis 1 and meiosis 2, and produce four sibling cells. The first phase of meiosis is interphase, and next, prophase 1. At the onset of meiosis, DNA strands thicken into chromosomes, homolog, or like chromosomes begin to approach each other. Homolog chromosomes pair to form bivalent. The centrioles divide and move to opposite poles of the cells. The bivalents duplicate to form tetrads or four chromatids group. The nuclear membrane disintegrates, crossing over, recombination occurs. In metaphase 1, the tetrads attach to spindle fibers at their centromeres line up at mite cells. In early anaphase 1, the tetrads separate and the paired chromatids move along the spindle to their respective centrioles. In late anaphase 1, the chromatids have almost reached the spindle poles. The cell membrane begins to constrict. In telophase 1, nuclear membrane encloses the separate chromatid. The cell membrane completes its constrictions. The first meiosis divisions end. There are no two cells each with the same number of chromatids as the parent cells. Prophase. Prophase 2 begins in the second meiosis division. Homolochromatids do not duplicate but merely separate. In metaphase 2, the chromatids line up at the mate cells, centriole at aster, and are at the poles. A spindle has formed. In anaphase 2, the now separate chromatids approach their respective poles. The cell's membrane begin to constrict. Telophase 2 has been complete. There are now 4 cells, each with half the number of chromosomes of the parent cells. This is a picture of the process of spermatogenesis. Spermatogonium is haploid, diploid, and then mitosis divisions, and primary spermatocyte, diploid, and in first meiosis divisions, secondary spermatocyte is haploid, and meiosis divisions 2, and then spermatids and haploid, and mature sperm cells is haploid. This is a picture of process oogenesis. Here, meiosis 1. After puberty, primary oocyte complete meiosis 1, which produce a secondary oocyte and a first polar body that may or may not divide again. The secondary oocyte begin meiosis 2, and meiosis 2 is secondary oocyte is haploid, and then ovum is haploid, and is three polar bodies. That was an explanation of cell divisions from me. Thanks for watching. Wassalamualaikum warahmatullahi wabarakatuh.